Okay, I think we'll go ahead and get started. Welcome to everyone. Good morning, good afternoon, and good evening, wherever you may be. My name is Rob Burgess, and I'm Chief Business Officer for the company Sino Biological. And I want to officially welcome each and every one of you, as well as our speaker, to the next installment in the Sino Biological sponsored and hosted webinar series on COVID-19 immunodiagnostics. Just a few housekeeping issues. All of the attendees, except for our speaker, will be muted throughout the webinar, but you can ask questions in the chat box. And I highly recommend that all of the attendees use the chat box to type in your questions. At the end of the webinar, I will select questions from the chat box and try to get to everybody's, and I'll ask our speaker to answer those. In addition, I want to emphasize that all of the attendees today will get a link later to a recorded version of this webinar. So be patient on that. And so without further ado, it is my pleasure and my honor to introduce to all of the attendees today our speaker for today's webinar. His name is Dr. Bobby Brooke Herrera. And Bobby is the co-founder and interim CEO, as well as Chief Scientific Officer for E25 Bio. E25 Bio is a spin-out of MIT, and the company is focused on developing rapid diagnostic tests for epidemic fever-related viruses, which include dengue, Zika virus, chikungunya, and SARS-CoV-2. Dr. Herrera is also a visiting scientist in the Department of Immunology and Infectious Diseases at Harvard. So Dr. Herrera, we are honored and delighted to have you here today to give this webinar. And with that, I will now allow you to share your screen and begin your presentation. Bobby, if you can, please.
Hello.
Thank you.
There you go. Hello. Thank you to Sino Bio for allowing me to come and talk about the important work that we are doing at E25 Bio, and thank you for the kind introduction, Rob. As he had mentioned, my name is Bobby Brooke Herrera. I'm interim CEO at E25 Bio, as well as a visiting scientist at Harvard T.H. Chan School of Public Health. Today, I will be discussing the development of a direct test, and later in the talk, I will discuss about possible implementation strategies of this kind of rapid test in a large setting dealing with COVID-19 transmission. As I had mentioned, the agenda, it's broken up into two parts. The first part of my talk will focus on development and evaluation of the direct antigen rapid test that E25 Bio has ventured into producing. And then the second part of my talk, it's a modeling study. This is purely a mathematical modeling based study, which shows the impact of large-scale surveillance on the COVID-19 pandemic, particularly in one region in the United States. This goes without saying, however, COVID-19 is obviously a worldwide pandemic. It's infected individuals in almost nearly every country on almost every continent across the globe. To date, August 4th, 2020, this screenshot was taken at 8:42 a.m. showing the total cumulative cases of COVID-19 has surpassed 18 million. I also show the top 10 countries which have experienced COVID-19, with the United States leading the bunch at nearly 5 million cases. As we all know, that the number of cases is also likely much more than 18 million worldwide, as diagnostic testing has been limited across not only the United States, but around the world. And so it's safe to assume that the case numbers of COVID-19 is much larger than 18 million. As an intro slide, I just showed this to introduce the way in which COVID-19 infects humans. While it's currently under investigation, a number of studies have demonstrated that COVID-2 originated from other coronaviruses that are primarily transmitted through bat populations. There have been a number of other studies that showed that it's possible that SARS-CoV-2 has also an intermediary hosts, other mammalian hosts, although there is strong evidence to suggest that CoV-2 originated from a bat coronavirus. Coronavirus enters the respiratory tract through many of the mucosal surfaces, including the eyes, mouth, and nose, and it goes into when it goes into the lungs. The spike proteins, which are the surface proteins on the surface of the virion, shown here, those surface spike glycoproteins interact with host cellular proteins known as receptors. In this case, a number of studies have strongly shown that SARS-CoV-2 enters through a receptor known as ACE2 or ACE2. Once it's inside of the cell, once it attaches to ACE2, it causes membrane fusion. It allows the virus to go into the cell and cause infection. A number of vaccine companies, as well as antiviral companies, are targeting spike glycoprotein on the surface of the virion because it creates an immunogenic response. And so should these vaccines, should these antibodies, should these antivirals target spike in a particular way, they have been shown to be effective in controlling COVID-19 infection in non-human primates, as well as now, as we all know, there are currently a number of phase 1, phase 2, and phase 3 trials that are ongoing, and we're hoping for a vaccine by the end of this year or early next year. Now, way of introduction, I will start to talk about the work that E25 Bio has been focused on. As Rob had mentioned in the beginning of the talk, E25 Bio is an MIT spin-out company co-founded by Professor Lee Gerke, as well as Dr. Irene Bosch and myself. Prior to COVID, we developed rapid diagnostic tests for several epidemic fever viruses, including dengue, Zika, and chikungunya. We pivoted our technology at the start of the year when we learned about the outbreak, and we tried to make an impact on the COVID-19 pandemic. So this first part of my talk will focus on the development of our direct antigen rapid test. As part of our platform technology, we develop monoclonal antibodies. There's a number of ways to develop monoclonal antibodies. However, one way in which we develop these monoclonal antibodies is by injecting mice with spike proteins. In this case, we injected mice with SARS-CoV-2 spike glycoprotein, and we fused these cells from the spleen or lymph nodes with myeloma cells, and these created hybridomas. Hybridomas are essentially cells that produce antibodies, or IgGs, as we call them. And what we did in this experiment, although it's hard to see, the point is not that I want you to read what's on the slide, but the important point here is that we're screening those hybridoma supernatants across a variety of coronavirus antigens or coronavirus spike proteins, including SARS-1 and SARS-2, as well as other related coronaviruses. And what you see in orange here is that we are getting recognition of our mouse hybridoma supernatant across SARS-CoV-2 in orange, but also a couple of other hits to known coronaviruses. We next take this through a proprietary E25 Bio high throughput screening platform. This is where we're taking combinations of monoclonal antibodies that we purify out of these hybridoma supernatants, and we pair them. And when we pair them, what we're looking for is detectability of a certain antigen. In this case, again, on the left side, we see a combinatorial matrix of monoclonal antibodies, pairs of monoclonal antibodies that are screened for detectability or for recognition of SARS coronavirus spike. You can see that in this matrix, there were a number of hits. Through further characterization, we identified a pair which we took further in our development. On this right side of this slide, we see a rapid test. In this case, it's a prototype test that we develop in the E25Bio laboratory. In this case, you see the control line, or C, which validates the test. And we're performing a limit of detection analysis to see how low we can go to detect SARS-CoV-1 and SARS-CoV-2 spike glycoprotein. We see strong detectability at 250 nanograms per mil on our prototype rapid diagnostic test. And we see detectability, although it's hard to view in this image, we see detectability all the way down to 1.98 nanograms per mil. This means that this is a good indication that we have developed a rapid test that would be within diagnostic range for detecting COVID-19 in humans. To further characterize our monoclonal antibodies, we performed additional analysis to start to understand where these monoclonal antibodies are binding. In this case, I'm showing Western blot analysis in which recombinant antigens from Sino are run on an SDS page, and we're using our candidate monoclonal antibodies. In this case, I'm calling them monoclonal antibody 16, as well as monoclonal antibody 18. And when you run a Western, we see that they have preferential recognition of the receptor binding domain. Both antibody 16 and antibody 18 have preferential recognition of spike within the receptor binding domain, although they do recognize to a lesser extent also subdomain 1 or S1 and S2. On the right side, this is just to show that control levels of antibodies that we inputted into the Western blot or into the gel, we ran equivalent amounts of protein. And so despite that equivalent amounts of protein were run on the gel, we see preferential recognition, particularly within the receptor binding domain. To further characterize our monoclonal antibodies, we performed linear peptide mapping. And when we do this in an ELISA format. However, we had clues that our monoclonal antibodies, again, targeted the receptor binding domain, which is highlighted in blue, in light blue here. And in dark blue and purple, we show the specific linear epitope, which is recognized by monoclonal antibody 16 and monoclonal antibody 18. What's important to note here is that I'm showing a sequence alignment of both SARS-CoV-1 and 2 in this figure. And this might explain why we see cross-reactivity between SARS-CoV-1 and SARS-CoV-2, but not other coronaviruses. And so this is further demonstrated in these tests. While I had previously shown you rapid diagnostic tests that were produced by E25Bio as a prototype, I am now showing you, in collaboration with one of our manufacturers, a manufactured dipstick that we call the Spike DART. Again, DART stands for Direct Antigen Rapid Test. These rapid tests are paper-based tests that provide a visual result in 15 minutes or less, and they can be scaled to millions per month. And so we view this as a really important way to control the epidemic. However, again, we manufactured our paper-based strips, and we took these into further characterization. Rather than running recombinant proteins from SARS and other coronavirus other coronaviruses, we opted to go into a high containment laboratory in which instead of running recombinant proteins, we're running live virus through our manufactured spike dart dipsticks. What I'm showing here on the left is you have a family of coronaviruses. The first two are the SARS-CoV-2 and CoV-1. The next three are MERS 229E and OC43. Again, these are related coronaviruses. And what we see here is that on the test line, so in the T region, we see detectability of both SARS-CoV-2 and CoV-1, but we do not get cross-reaction against MERS 229E and OC43. In further characterization, as we did with the recombinant proteins, we also ran a limit of detection study using live virus. In this case, we're running SARS-CoV-2. And what we see is a nice degradation of detectability, in which we see strong detectability when we're running 10 to the 5.4 TCID 50 per mil. However, we lose detection between 10 to the 3.1 and 10 to the 3.8 TCID 50 per mil. Ultimately, what this means is that we are in diagnostic range for detecting CoV-2. We have confirmed that we detect on recombinant proteins. We have further confirmed that we detect to low limits of detection using live virus. And we know now that our rapid paper-based strip does not cross-react with other common coronaviruses, such as MERS 229E and OC43. Finally, we took this to validation. E25Bio has performed one of the only prospectively collected clinical studies in the United States, as well as we have done retrospective testing on our manufactured spike dart dipstick. In this case, I'm showing data from one of our retrospective analysis from individuals infected with COVID-19 in Brazil. And what's important to highlight is a couple of things on this table. First, I'd like to talk a little bit about our population. The number of cases involved in this study was 121 total individuals that we evaluated with the spike dart dipstick. Of those 121 patients, 72 tested positive by gold standard polymerase chain reaction. Again, PCR, the gold standard, is a nucleic acid-based approach that is that contains high sensitivity, although there are caveats to PCR such that it has a reduced ability to scale across systems because of the complexities in involving a laboratory-based instrumentation and trained personnel to perform these tests. In addition to our 72 positives, we also evaluated on 49 individuals who tested negative by PCR on the for COVID-19. A really rigorous way to do analysis is also to do a distribution across cycle threshold values, which you see up top. Lower cycle threshold values between 5 and 20 indicate that an individual is early in the phase of infection, meaning between 1 and 7 days of symptoms onset, you would expect an individual to come up by PCR with a CT value between 5 and 20. Beyond 20, these individuals are getting into the later stage of their disease course, of the COVID-19 disease. And so therefore, you would expect lower levels of antigen in these populations of patients. In any case, our overall sensitivity and specificity is quite high. I show it here in red, where the overall sensitivity or percent positive agreement is 84.7, while our specificity or our percent negative agreement is around 86%. And you can see, again, the distribution of sensitivities and specificities across the distribution of PCR-CT values, where in individuals, again, at high viral loads, meaning these individuals are in the early onset of disease course, in between days 1 and 3 of infection, you see that the sensitivity increases dramatically beyond 85%, well above 90%, and even in some cases to 100%. Before I talk about our next phase of the talk, I just want to summarize again about the E25 Bio direct antigen rapid test for COVID-19. Again, this is a simple paper-based strip that provides a visual result in 15 minutes or less. We can manufacture these tests, and we're currently manufacturing these tests at 2 sites, one in the United States, another is in an international site. Between these 2 sites, we're able to scale production of our rapid tests to the millions per month. And we truly believe that rapid testing can impact the way that we respond to the COVID-19 pandemic, and ultimately the way that we can control the COVID-19 pandemic. And so E25 Bio has been involved in a number of modeling studies, one of which I will present today. And it's just a couple of slides which I'm showing, which I'm illustrating the impact of rapid testing on the COVID-19 pandemic. We adopted a previously developed enhanced SEIR model to evaluate the impact of COVID-19 surveillance. We enhanced this previously developed model to consider 8 states shown here, where you have a state of quarantine and uninfected individuals, although these quarantined uninfected individuals can become susceptible and therefore either infected and undetected by PCR or infected and detected. These individuals that are infected and either undetected or detected can either go to a recovery state, where these recovered individuals will enter a quarantined state and ultimately revert back to the recovered state. However, there's a certain set, as we know about the COVID-19 pandemic, is that there's a number of infected individuals that have the probability of becoming hospitalized and experiencing severe disease. And so we have a state here where you have these infected individuals going into a hospitalized state. These hospitalized individuals are either recovered, they either recover, or unfortunately, as we have seen across the globe, a certain number of individuals succumb to disease and are therefore categorized as extinct or deceased. I just want to highlight here these points in case there are any questions at the end, but gamma and psi correspond to the rate of individuals entering and exiting this quarantine state. Beta corresponds to the transmission coefficient. Epsilon and nu correspond to the rate of detection. Lambda, rho, and sigma, which you see here, those correspond to the rate of recovery for the infected, detected, and hospitalized. Mu is the probability of developing life-threatening symptoms and therefore being hospitalized. And tau is the rate of mortality, or the mortality rate. So we applied this, this enhanced mathematical modeling scheme to try to understand implementation strategies in certain regions in the United States. And in this particular presentation, I'm showing our work in California. So California is amongst one of the states in the United States that has seen a tremendous number of COVID-19 infections. The total infections to date is 514,000. A majority of those are clustered within the Los Angeles County region. By implementing a COVID-19 strategy, or we sought to evaluate the hypothesis of what would frequent testing using rapid diagnostic testing do to a number of the states in our model. And so what I'm showing here are 3 states, although of course happy to talk a little bit about the other states, but I'm focused in on the undetected infected, detected infected, as well as our individuals that succumb to disease. And so what we see here is, based on our modeling, is that by implementing a rapid testing surveillance strategy in Los Angeles, and performing rapid diagnostic tests every day, every 3 days, 5 days, 7 days, 10 days, or 14 days, you see the transmission, or you see the percent population of infected individuals decreasing dramatically as compared to the 2 blue lines, which show that these otherwise undetected infected individuals continue to increase as we currently are experiencing. The same is the case for the detected and infected. In this case, these individuals are being detected as infected. However, what we show here is that rapid testing, frequent rapid testing between every single day rapid testing, as well as in some cases every week or every 2 weeks, in the case of the green and orange lines, we see that frequent rapid testing ultimately would be able to reduce COVID transmission in Los Angeles. Finally, I show what is the impact of rapid testing on the number of people dying. And it makes sense if we're showing here that the number of individuals who are either undetected and infected or detected and infected with a rapid diagnostic testing strategy, a frequent rapid diagnostic testing strategy, as those numbers of infections decrease, ultimately what you're going to get, ultimately what will be achieved is a reduced number of deaths across Los Angeles County. And so ultimately what this advocates for is that not only in the United States, but across the globe, whether we should be able to implement a rapid diagnostic testing strategy that is frequent actually impacts the way that we control this epidemic. And I just want to highlight one more point here. Most of our models, while we have done testing across a number of testing strategies, what I'm presenting here is a testing strategy in which an antigen-based test has an accuracy with an 80% sensitivity and 90% specificity. So what this means is that despite antigen-based tests being less than perfect, for example, compared to gold standard PCR, we are able to impact the way that we control rapid transmission of COVID-19. And we're using real data collected off of government data released in California to show this modeling data. Finally, in my last slide, I wanted to present a further rapid diagnostic testing surveillance strategy. In this case, we're honing in on the diagnostic testing strategy within California. And particularly here, again, our sensitivity and specificity is 80% and 90% respectively. However, despite this accuracy level, by honing in on high transmission regions, increasing the frequency of testing in high transmission regions in California, such as Los Angeles, and decreasing the frequency of rapid testing in lower transmission regions. As we had seen, there are certainly lower transmission regions in California. We're still able to reduce, as we see here in this darker blue line, you're reducing the number of the percent of the population that is infected by COVID-19. And this is purely done by a testing strategy in which we're selectively targeting higher frequency of testing in regions that are experiencing high transmission of COVID-19. So this is one way to selectively target regions, whereby also reducing costs, in which you don't necessarily need to do daily rapid testing in all regions, but rather focus on select regions that are experiencing high transmission of COVID-19. So in summary, of course, I'd like to give acknowledgements to many of the individuals that have not only contributed to development of E25Bio's direct antigen rapid test for COVID-19, but also to the number of individuals that have contributed to our work on the modeling side. And so on the E25Bio side, again, special shout out to our entire laboratory staff, in particular, Irene Bosch, Noel, Adam, and Ankita. I'd like to give a special shout out to our COVID mathematical modelers, Bia and Anthony and Miguel, our collaborators at MIT and Tufts, Professor Lee Gerke and Elena Nemova, and then of course, Sino Bio. As we all know, Sino has been instrumental. Sino Bio has been instrumental in providing not only E25Bio, but the world with reagents to help launch development of not only rapid diagnostic tests, but also vaccines. And so we thank Sino Bio for being a great partner to E25Bio. So with that, I'd like to open up the discussion to questions. And then of course I put my contact information for those who may want to follow up with a question later on.
Thank you, Dr. Herrera. That was an excellent talk. We sincerely appreciate your time today. It's very enlightening to see the twist on live viral detection via antigen, as opposed to detecting, for example, antibody response, like the detection of IgG and IgM. So let me go ahead and start. I'll ask the first question. I guess it's my question here, and that is, can you tell us a little bit about the advantages to detecting for the presence of the antigens in the live virus, as opposed to detecting for the presence of an immune response via the presence of IgG and IgM?
Sure, and thank you for that question. I think ultimately that's a very important question for the audience to understand, and this goes back to the importance of developing a direct antigen rapid test. And so when an individual is infected with SARS-CoV-2 and is experiencing COVID-19, again, the disease course is quite prolonged, meaning in the first phase of the disease, in the early onset of disease, between 1 and 7 days of infection, there are really two components, or two viral agents in which one is able to detect. That's either nucleic acid through PCR or antigen-based testing through antigen diagnostic tests. Later in the disease course, one starts to develop an immune response. By day 10, by day 30, as we've heard around, individuals are now mounting robust antibody responses. And so while an antibody-based test might tell you that you were previously infected, they do not tell you that you are actively infected. And so one importance here, again, that I would like to highlight is that an antigen-based test allows individuals and allows their healthcare providers to identify individuals that are experiencing active COVID-19 transmission. And therefore, as we see in our modeling study, you then are able to isolate those individuals and ultimately quarantine them such that those individuals are no longer transmitting virus and disease to other individuals.
Got it. Great. So it's really a two-pronged advantage there. It's about active infectivity, and it really is focused on the earliest onset of infection, right? Even before you see the presence of the immune response.
That's correct.
That's great. That's great. So I'll just get into some of the excellent questions we've gotten from our attendees. Can you just comment on the detection of the spike protein versus the N protein, and why someone would choose spike over the nucleocapsid protein?
Sure. I think that's a fantastic question. A number of rapid diagnostic test manufacturers have focused on nucleocapsid. Nucleocapsid, while I didn't mention it in my talk, is a surrogate antigen that is encased within the virion. And ultimately, you are able to lyse open that virion and release nucleocapsid proteins for other manufacturers to detect. And certainly there have been a number of antigen diagnostic test manufacturers that have received, in the case of the United States, approval for use of their test. However, in addition to nucleocapsid, there is also the spike protein, which again sits on the surface of the virion. It's a surface exposed protein. And so because of that reason, it is abundantly expressed on the surface of the protein. It allows us to detect live viral particles or live virus itself. And so therefore, there's some of the advantages of going after the spike protein. And to my knowledge, I know that we're one of very few companies that has focused our rapid diagnostic test development towards detecting spike protein. Now, whether there are advantages over one of the other, I haven't really seen advantages in terms of our accuracy levels. We're right in line with many of the other nucleocapsid-based antigen tests. And so it's just an alternative way. But the important point here is that both spike and nucleocapsid are abundantly expressed and released into respiratory samples where one would be able to detect COVID-19 infection.
Sounds great. Sounds very good. It's very logical. And I'm getting a number of questions in the chat box on sample type compatibility. And I'm just wondering if you can comment in your data, was that whole blood, was it serum, and are there other samples for which your test might work, like sputum or even saliva or a nasal swab?
Sure. Our rapid diagnostic testing has been robustly evaluated on the standard, which is a nasal pharyngeal swab. The data that I presented today was collected on nasal pharyngeal swabs. However, we have done other testing on nasal swabs as well as serum. And in these, as well as saliva, so nasal swabs, saliva, and serum. And in these sample matrices, we have been able to detect COVID-19 using our spike DART dipstick as well. And so we hope that in the future, while our current version of the test is more tailored towards those respiratory, nasal, pharyngeal, and nasal swabs, we hope then we can expand the testing platform by incorporating other sample matrices, which include the less uncomfortable samples, I'll put it that way,
Right.
by going after serum or even saliva.
Yeah, well, that's fantastic. It sounds like your sampling population, the types of sample that you're collecting are relatively non-invasive, which makes your system very adaptable, I think, worldwide. You had mentioned the accepted diagnostic range for detecting antigens and I guess live virus. How did you define this, or how did the research community or the diagnostic community define what this range is?
Sure. I think also that's a really poignant question, especially for E25 Bio in particular, as we're trying to get our dipstick validated by the FDA in the United States. The FDA at the early onset of the pandemic put out policy that suggested the accuracy level for an antigen rapid test should be around 80% sensitivity. And of course, this is in comparison to polymerase chain reaction. So it's a bit confusing why they would do that. One, because you're comparing apples to oranges. You're comparing an antigen test, antigen detectability, to nucleic acid detectability via PCR. And so in my opinion, I think that that was an unfair advantage to the antigen test manufacturers, such that we had to compare against PCR. And really, I think where that metric or where that standard came from was from previous experience on influenza virus. So as we all know, the world has experienced a number of influenza viruses. For example, H1N1, which was a quite dramatic epidemic around the world. And during that time, they had set a standard of 80% accuracy. So I think they were going off of that level I think they were going off of their experience, which is why they implemented that 80% sensitivity threshold. However, what I will say is that, and I did not show it in my modeling presentation, although we do show that even with frequent rapid diagnostic testing, in tests that contain lower than 80% sensitivity, you still dramatically reduce transmission of COVID-19. Even in rapid tests with 60, 70, 75% sensitivity. And so this is part of the public outreach that E25 Bio and several of my other colleagues are trying to do, is trying to encourage FDA and other regulatory bodies to advocate for rapid diagnostic testing strategies rather than solely focusing on gold standard PCR.
Great. Thank you for that excellent answer. And I've got another good question here. Do you think it's a good idea to combine your antigen type testing with more conventional antibody testing to improve accuracy? And the attendee cited that actually happened with HIV. Is that necessary?
Of course, I don't want to downplay any type of testing strategies. PCR is very important, as is antigen testing, as is antibody. All three forms of testing is very important in controlling the epidemic. I want to first start by saying that I think frequency of an antigen test is actually very important to increasing the level of sensitivity. So again, rather than using an antigen test weekly, if you, and our modeling data shows this, rather than doing one rapid test every week or every 2 weeks, how about doing it every 3 weeks or every day? Because of the dynamics of COVID-19 in patients, the dynamics of the virus, the dynamics of antigen presence in the system, if one is able to increase the frequency of an antigen test, you will likely increase sensitivity of that particular antigen test. This is something that we've also shown in a number of studies, not for COVID, but for our other disease indications, such as dengue, Zika, and chikungunya. But moreover, to the point of the question, I also want to say that a combined strategy of antigen and antibody testing could increase the value of diagnostic testing, in which of course, you're following an individual throughout the course of disease, of COVID-19 disease, in which you're using an antigen test to try to identify active infections, and then use antibody tests to identify those who were previously infected. Either IgG or IgM antibodies can be used in this case. And so I don't want to necessarily say that a combined antigen plus antibody strategy is not okay, because I also think that that is a good way. Although, again, when you're detecting antibodies, individuals are likely no longer infectious. And so they have gone through the infectious cycle, infected many others around them. And it's rather just an immunity passport, just to let individuals know that they were previously infected.
Right. Great. Yeah, so and also I think there's a macroeconomic implication there too, is it's just much more expensive to do two different types of tests on a single individual. And at some point you have to make a difficult decision on what's the best test for that particular person.
That's right.
So a question from India, and I know you had a full slide on this, your slide about California and how early and rapid testing really does prevent spread of the disease. But in India specifically, I mean, can you elaborate a little bit more on that slide about how you would see rapid testing for the antigens and the live infectious virus would help to control the spread?
Sure. While I presented data on Los Angeles,
Right.
I've also extended our model to a number of other states focused in the United States, although I truly believe that this broad implementation model could be scaled across other states in other countries. And so again, what the point that I was trying to show here in our modeling data is that it doesn't really matter the state. In this case, I'm using Los Angeles as an example because the data being collected in Los Angeles, as well as New York and Massachusetts, other data sets that I'm using are quite robust, and it allows us to have access to a lot of this data that other countries do not provide. But in any case, what I'm showing here is that frequent rapid testing dramatically decreases transmission, or the percent of the population that is actively experiencing COVID-19 disease. Subsequently, when you reduce the percent of the population that is infected, of course, you're also going to reduce the number of people dying. And you see that here, whereas this is the percent of the population that is experiencing death, versus the two blue lines in which you're not really performing a testing strategy, in which, of course, the deaths continue to increase. And this is using, again, real-world data collected from the Los Angeles government public database. However, we truly believe that this implementation strategy could be scaled not only to states within the United States, but to other regions throughout the world.
Sounds great. Thank you for that answer. And I think we're really starting to hit home on the business model and I guess the financial model. A number of individuals have asked, what's the cost for like a single spike dark dipstick test?
Sure. Of course, you know, E25 Bio is a United States-based company, although we have global aspirations. And one of the important points about our types of testing technology, which is essentially a laboratory on a paper-based strip, what that ultimately allows us to do is significantly reduce the cost of our test to the end user. There are certainly a number of antigen-based tests that have come to the market. These antigen-based tests require an instrumentation to read the result. And when you add in the requirement for an instrument to provide the diagnostic result, then of course you have manufacturing limitations in which now you're not only manufacturing the rapid test, but you're also manufacturing the reader to read the rapid test. Of course, when you're introducing that type of system, the cost is going to increase. And so antigen-based testing technology is much more cost-effective than gold standard PCR. And then moreover, a point further, because E25Bio does not, it's a paper-based test that does not require any instrumentation to provide the visual result. Therefore, we're able to decrease the cost even further. And so we hope that our affordability can be scaled not only in the United States, but we want to make it as part of our mission that our tests can be sold in other countries in Latin America, in Southeast Asia, in Africa, etc. And so we aim and we intend to produce affordable rapid diagnostic tests.
Great. And related to that, I have to ask, and if it's okay for you to answer, what is the status of your regulatory filings for these dipsticks, and are they already approved maybe for CLIA usage, or what's the timeline?
Sure. So as I had mentioned, E25Bio was one of few companies that decided to go down a prospective as well as a retrospective clinical study. And because of that, it delayed our regulatory filing. However, we learned so much about our diagnostic test, as well as, in my opinion, it has been the most rigorously validated because we did it on a prospective collection. Right now, our FDA regulatory filing is pending, and it's pending submission of our final clinical data. Our timeline for launch, E25Bio is really hoping to launch this test for use not only in laboratories, but also at homes, across workplaces, at schools, and across the globe as soon as possible. And really, we hope to achieve this within the month of August to early September.
That's great.
Of this year.
Very exciting. So I think we're now just about out of time. We'll finish with that question, but Dr. Herrera, I want to congratulate you and the others at E25Bio for an astounding technology. It's extremely exciting. And we at Sino Biological appreciate E25 as a partner, and we wish you the best of luck in your regulatory filings and executing your business model worldwide. So we're very pleased to be associated with your company, and we really appreciate your time today giving this webinar.
Well, thank you so much again for the time, and thank you to the audience for tuning in and learning a little bit about what we're doing at E25Bio.
Yep, and let me thank the audience as well. I appreciate all the attendees. We had great turnout today. And thank you to the attendees for the excellent questions that you asked. I apologize for not getting to every question, but we had a time limit here. Please tune in, check out the Sino website, and register for our remaining COVID-19 immunodiagnostics webinars. And on a final note, I just want to thank my colleague and good friend, Dr. Jen Liu, who actually executed and helped to host this webinar today. So with that, I will sign off, and I will say good morning, good afternoon, and good evening, wherever you may be.